discuss today in this panel is very relevant as there has been so many advancements in the field of therapeutics, and it's a pleasure to introduce and invite the esteemed scientists to discuss the topic of next generation therapeutics, from whom we will be hearing today the lessons from their journey, challenges, and way forward. The field of therapeutics is rapidly evolving with the breakthroughs in the gene therapy, personalized medicine, and biologics reshaping the way we approach treatment modalities. These advancements offer unprecedented opportunities to address the complex diseases that were once thought unrealistic and uncurable. I would now like to invite our speakers on the dais. So please welcome Dr. Ajit Kamath, the moderator of the session. Dr. Kamath has more than 30 years of experience in preclinical drug discovery, primarily at Pfizer Worldwide R&D, US, and head of the external R&D innovation Pfizer in India. He has worked in Jubilant Biosciences and Mitra Biotech. Currently, Dr. Kamath also serves as an advisor to Pandorum Technologies and Mesastop Solutions. The next speaker is Dr. Murli Ramchandra, CEO of Origin Oncology Limited. He has contributed to the identification of 20 innovative drug candidates which are undergoing global development and one approved gene therapy for bladder cancer. Dr. Narula is Dr. Gaurav Narula is a professor in pediatric oncology and health sciences and senior clinical lead at Tata Memorial Center, Mumbai. His research is focused on improving the outcomes with the innovative approaches, including the low-cost alternatives, improving supportive care using innovative technologies such as CAR T cells. Mr. Sanatan Upmanyu is the head of data sciences and executive director at Invent AI, and he led the development and deployment of AlphaMed platforms, a cutting-edge AI system that accelerates the drug discovery and healthcare innovation. Please welcome. Dr. Nitin Damble is Chief Innovation Officer at Sun Pharma Advanced Research Company, and so has more than 25 years of experience in exploratory biomedical research, drug discovery, preclinical development in oncology and inflammatory diseases. Dr. Sridhar Narayanan, a passionate drug hunter, has more than 20 years of experience in drug discovery and development. Dr. Narayanan is the founder director of FNDR. Welcome, sir. Dr. Anna Visala, Joint Drug Controller at CDSCO and Directorate General of Health Sciences. Ma'am, in her current role at CDSCO, focuses on critical areas such as regulation of the recombinant products, investigational new drugs, and cell and gene therapy products, and assists the Drug Controller India in enforcement of the Drugs and Cosmetic Acts. Let's give a warm welcome to all the speakers. So as we begin this session, I would like to highlight the few outcomes of what BIREC has been doing in new generation therapeutics. As you can see that, BIREC supports the innovative projects in the area of biotechnology, starting from ideation to commercialization stage. And these are divided in various thematic areas. And next generation therapies are mainly supported by BIREC within the various segments through different programs, which are designed to support the various products at different stages. And in this area, the total project cost is nearly 105 crores, which has been supported, where BIREC has contributed nearly 110 crores. And here are the few highlights where there are successful project highlighted, where Urigro is the India's first urology cell therapy product and FDA approved therapy for the treatment of the urethral structure is commercialized. Development of CD19 CAR T cell therapy for the relapsed and refractory B cell non-Hodgkin's lymphoma has successfully completed phase 3 clinical trial. The bench to bedside development of the first in India CAR T cell therapy was supported for phase 1 and phase 2 clinical development. Clinical trials are underway for allogenic cell therapy products for the treatment of dry age-related macular degeneration and indigenous autologous anti-CAR-19 CAR T cell therapy for CD19 positive lymphoblast leukemia and B cell lymphomas. The clinical studies are ongoing. So BIREC has not only supported the treatment modalities, but also the facilities and the capacity enhancement where it has the cell integrated cell therapy development and manufacturing facility has been supported, and lentiviral manufacturing facility for manufacturing of CAR T cells and conducting the clinical trials has been supported. So with this, we will have an exciting session from the speakers who are in the field. And I would like to invite Dr. Ajit Kamath, who is the moderator for the session, to please start the session, sir. Thank you and welcome.
Yeah, thank you, Dr. Aparna. Thank you, Bairak, for this wonderful opportunity to be a part of this esteemed panel. Okay. As Aparna mentioned, I think lots of exciting work is going on next generation therapeutics, and we are trying to, through this panel today, we are trying to showcase some of the examples to you as to what the opportunities are there and how India can prepare for next generation therapeutics delivery for our patients. I'll quickly, to set a context for this particular presentation, I'll quickly take you through as to the progress that has been made in this particular area in the past few years. If you look at the various drugs that are approved in the last 30 years, most of them have been focused on small molecules. You can see the trend in this particular slide going up to, 30 years ago, perhaps 90% of all the drugs were small molecules. And now you fast forward 2023, you see about 40% of them are non-small molecules. They're all biologicals. And that's where I think, India can play a bigger role going forward, not only in supporting the generics where we are the leader, but also looking at how can India contribute in developing novel molecules, but also support biosimilars area. Okay. So if you look at the opportunities within biologics or within non-small molecule area, you will see this is the type of molecules that are there that are approved in 2023 by the US FDA. While 60% of them are all small molecules, you see large number of opportunities in oligonucleotides. You see opportunities in hormones, looking at bispecific antibodies, looking at monoclonal antibodies. So the share of biologics is going to grow as we go forward. Currently, it's about 40% 40% biologics, and the remaining have been non-biologics. Okay. The opportunities for generating next generation products are tremendous. This is one example as to how the field has grown. It hasn't grown as much in India. We'll discuss that in our session today. But if you look at how things have progressed worldwide in the last 10 years, tremendous progress, about 4 to 5-fold increase in the number of projects, what we call it as cell and gene therapy. In this particular area, it will be on RNA vaccine, DNA vaccines, RNA therapeutics, CAR T therapy, and of course, gene editing and other spaces. So this field is really growing, and we would like to be a part of this particular field. Cell and gene therapy, you may not be able to see the slide, but just see how fast it has grown in the 10 or 15 years. Number of projects that are in the cell and gene therapy and the opportunities that are there to cure diseases that cannot be managed with small molecules. The diseases are getting complex, so you'll have to have a better way of treating diseases, treating unmet medical need, and this would be one way of approaching, that is with the cell and gene therapy. It's about 10-fold increase is what is expected for FDA approvals between now, between 2017 and 2030. That is tremendous. It's 10-fold number of more FDA approvals. On the right-hand side, you will see the amount of revenue that you can generate. Again, about tenfold more. Instead of 5 billion, now it is going to be 60 billion in about 2 to 3 years. So there is highlight, it's important to know how valuable these tools are to treat diseases that have been not addressed so far. It's not only a question of, how do we treat the disease, but also a question of what are the various modalities that are there. And you can see this particular slide from pharma projects. Okay. While small molecules has been the bread and butter for most of the pharmaceutical companies, you can always see the number 2 to number 10, they're all on biologics, either antibodies, monoclonal antibodies, bispecific antibodies, various cell-based therapies, stem cells, RNA therapy, and everything else. This is just to tell you the opportunities that are there that you can use to treat diseases that have not been able to manage with small molecules. On the right-hand side, you see the effort that is going on in smaller companies to address. As you can imagine, many of the startups always looking for a new way of addressing diseases, and that's where I think we had very good discussions yesterday as to how we can support the startups to look at diseases in a different way and come up with treatments. And you can see this particular slide where bigger pharmaceutical companies, which are focused on diseases that will affect larger population, they're also trying to do some work internally, but mostly everything happens with partnership. And you can see the merger and acquisition volumes here. Small molecules again lead, but antibodies, ADCs, biologics, and so many other areas, there are opportunities for partnership between startups, small companies, to partner with the larger companies to bring the molecules to much needed unmet medical need. In addition to the molecules that we talked about, cell-based therapy, gene therapy, also opportunities exist in oligonucleotides. You can see the slide on the left-hand side. You can see there were very few approved drugs, antisense oligos, perhaps about 10 years ago, 15 years ago, and you can see the trajectory, how many diseases are being addressed by antisense oligos. On the right-hand side, this particular slide tells you the synthetic DNA. Most of us researchers have been thinking about, making DNA using cells and purifying the DNA, but now you can go up to 10 kb of synthetic DNA. So think about the speed in which you can progress your project, in this particular area. So I just want to highlight some of these key things for our discussion today, and then end with a couple of slides. One is the modalities, just capturing all the various activities that are ongoing here in India, supported by BIRAC on cell-based therapy, CAR-T therapy, antisense oligos, mRNA. We see lots of startups working in this particular area, and I hope they can move the project to the next level and then eventually bring it to the patients. Today, we are very pleased to have an esteemed panel with variety of experience, and we are looking forward to the discussion today and your participation in this particular session. We have clinician Dr. Narula is here. Okay, he's working on a novel therapy. You will hear more from him directly, treating patients. He sees patients every day. Thanks for taking time to come here for sharing your experience. Then we have preclinical researchers. We have Dr. Damble, Dr. Murli, and Dr. Sridhar, okay, who have demonstrated experience like Aparna said, about 50, 60, 70 years of combined experience in doing drug discovery for novel drug therapy. And they'll be talking about their approaches in their companies to handle next generation therapeutics. And then we have Dr. Sanathan. He will be talking about AI/ML, the advantage of this particular platform for accelerating drug discovery and also for clinical trials. And then most importantly, we need a regulator. And we are glad that Dr. Vishala is here with us, who routinely reviews proposals and then provides guidance as to what type of documents are required, what type of studies are required in order to be approved by the CDSCO. So we are excited, looking forward to the discussion today and your participation. Over to you, Dr. Narula, if you can come over and do your presentation. Each of them will have about 8 minutes of presentation, and then we'll have a group discussion. Thank you.
Right. So you want me to plug it in? Right. Green red. Yeah. Yeah. Right. Thank you very much. Apologies for that slight hitch because there was some problem in loading it earlier and we had to do it directly over here. Uh, I'm a clinical oncologist, a medical and pediatric oncologist who works in one of the largest cancer centers in India and in the world. These are my disclosures. I will be talking to you about our journey through the first cell and gene therapy in India and how it started. For me, this personal journey started shortly after I left the Navy, where I was working as a hematologist about 12 years ago and joined Tata Memorial Hospital to work in the pediatric oncology department. A friend of mine sent this email with this link, and when I opened it, it was about the story which was on the cover of New York Times about Emily Whitehead, a girl who has received CAR T cell therapy. She was, at pretty much the end of the line. Her disease was refractory, not responding to anything. She had very little time left to live. And then she was given this new experimental therapy. She had a very dramatic response. She went into a coma with severe fever and hypotension, was on the ventilator. They discovered that it was due to very high IL-6 levels. They managed to give her tocilizumab, a specific IL-6 inhibitor, and she miraculously recovered. And 6 months later, her disease remained in complete remission, which was sort of unheard of in the kind of clinical situation she was in. Now, this was a very dramatic story, and the way it was done with this CAR T cell therapy, when I discussed with my friend, I remember his words, they stuck in my mind forever. He said, Gaurav, this is science fiction. And in 2012, really, it did sound like that. But this was with the MLE story, there was a rapid rise in interest in CAR T cell therapy around the world. And some of this was just shown to you by Dr. Kamath. And the slide over here shows you how there was an increasing number of trials across the world. And within a short span of time between 2012 to 15 to 18, China overtook India as a hub for clinical trials. And this map shows you clinical trials which were going on in CAR T cell therapy space across the world in just 2020. It has more than doubled and quadrupled from this figure by now. But as you can see, there was nothing happening in the global south. And sitting in 2015, we realized that this is never going to come to us if we keep waiting for it. Sure enough, by 2017, the first product which was used to treat Emily Whitehead was approved in the US. Cambria by Novartis took it over. The product was developed in the University of Pennsylvania and taken over by Novartis, went through phase two, got FDA approval, and within a short span of time, other approvals started coming, and now there are a host of cell and gene therapies now which are now approved across the world, with more happening every year. But along with the approvals came the price tags, and these price tags put them very much in the unaffordable range. $500,000 makes it beyond the range of most Americans and developed countries, in fact. So forget about low medical income countries like ours. In 2015, our struggle with ALL was very, very different from what Emily Whitehead's story was. This graph shows you the improving outcomes of childhood ALL with each passing decade, starting from the 60s. And you can see how the outcomes have been improving with each decade. And by the 2010s, and this is in the pre-CAR T era, the outcomes in the West and in developed countries have already reached 90 to 95%. This is with chemotherapy alone and salvage therapies like second line therapies and transplant. So with these outcomes, with these therapies alone, they have reached 95%. For them, CAR T cell therapy is a bridge to the last mile bridge into curing those few children out of every 100 who are not getting cured with currently available therapies. Whereas in India and like most developing countries, our outcomes for childhood ALL hover around the 60 to 70% mark. And this was the challenge that I was facing as a clinician in my clinic, sitting in Tara Memorial Hospital in 2013 and 14 and 15, when all this was going on around the world. With the time, our outcomes have also been improving. Now, this graph on the left shows you the outcomes of children in Tata Memorial Hospital, and you can see the blue bar is from the earlier era, the green bar is from the more recent era, and the outcomes have been improving. But if we have to jump from 70%, 75% to 95%, we are going to have to wait 4 decades, 5 decades to do it at the current pace. So, because when these children relapse, they do not do well at all. The next graph shows you the same children in this story who did not relapse, who did not whose disease did not respond or came back despite chemotherapy or transplant, did not survive at all. And this is our biggest challenge. For this, we first, before they say before you have a fancy car, you have to think of bread and butter. We have a foundation called the Impact Foundation, which supports the treatment of children at Tata Memorial Hospital. Regardless of paying capacity, any child who walks in, we are able to give them the full treatment, state-of-the-art treatment, right down to CAR T cell therapy if needed, by this extensive mechanism of support. And by 2014 to 15, we had a fairly good system in place where we could achieve this. It's run by my dear colleague Shalini Jatia, who helps us to manage this foundation. But clearly, this was not enough. And that is why in 2015, I met Dr. Rahul Purwar, who was working in IIT Bombay, who was also working on CAR T. And we got together and we decided that if he could develop a novel CAR T in his institute, we would be able to take it through clinical development and to clinical trial. We were partnered and mentored in this by scientists at the clinician scientists at the National Cancer Institute who guided us through this. However, I'll be very clear, we received neither funding nor any materials from the National Cancer Institute. It was just a knowledge partnership, and therefore this entire thing from beginning to end was made in India. These are the milestones of our journey. We started in 2015 when we met and TMC and IIT Bombay decided to collaborate. We formalized this into MOUs with ourselves and with National Cancer Institute. Initially, we were running around for grants. We worked with whatever small intramural grants that we could get. I must say our institutes backed us to the hilt, though even if at that time grant agencies were understandably very skeptical. Almost every grant application which I put was summarily rejected within seconds, I think. It just came back. But over time, we started getting some grants. We initial grant from Tata Trust really got us going, about 3.5 crores. That helped us to set up some facilities and work and complete the preclinical development. By now 2019, we were getting approvals from the preclinical work was in an advanced stage, and the IND application had been done. In March of 2021, we got the historic first approval from the CDSCO for first in human clinical trial on cell and gene therapy in India. And by June 21, 4 June 21, we were able to infuse the first patient and became a historic day for our country. By December 22, the phase 1 trials were completed and we got permission to start phase 2 trials. And on the basis of the adult trial, which got completed last year, on 13th October 2023, the CDSCO approved the drug for use in adults with relapsed and refractory B-cell leukemia and lymphoma, making it another historic day for our country. And ImmunoAct, which is a company founded by Dr. Rahul Purwar, were this approval, and this was a huge achievement for us. With this, but the journey through this regulatory compliance was not easy because when we started out in 2015, there was no regulatory framework for a CAR T cell therapy in India. We did not know what norms would be followed because they were not there. So this journey of the regulatory compliance also sort of evolved along with our journey. We initially benchmarked ourselves to US FDA, saying that whenever it comes, we'll be able to match it. And but every committee that we went to, and we had to go through a host of different committees, many of them which we did not know before this. But when we had to go to them, and they were also dealing with a proposal like this for the first time, everybody was very helpful and cooperative. But there was a long journey and path and resistance which we had to get through, and ultimately we got these approvals. This is the preclinical work was published, and the first patients were infused, and there was a lot of celebration around. And finally, I can say that we were on the clinical trial map of CAR T cell therapies across the world. And since then, it's not stopped since then. Initially, these opened as trials from academic institutes like IIT Bombay and Tata Memorial Center, which led the clinical trials. But then later on, ImmunoAct, founded from IIT Bombay, took over the phase two trials, and now they opened it up in multiple centers. Immunil, another company, started opening a trial in Bangalore, and then another trial opened from CMC Vellore. And as you can see, over a short period of time, the clinical trial scenario has lit up. Now, Dr. Reddy's has opened a trial for myeloma. So now the country's landscape has really lit up with cell and gene therapy, and we are very, very happy that this is happening. This is a snapshot of the results from the pediatric phase 1B trial. I'm sorry, the green bars do not show for some reason on the screen, but but this shows you the three dose levels that were tried. The yellow block is the initial dose levels. It's a phase one trial, so we start with the lowest possible doses and go up. The phase one trials, most patients had either a temporary response or partial response. But the moment we went to 3 to 5 million per kg and above, every patient had a response. And except for that one red bar which you see with a cross at the end of it, every other patient is in a sustained ongoing remission and close to 1 year of everybody has crossed 1 year by now in sustained remission, except for one patient who's reaching 1 year by next month. So the responses in BALL in children we are seeing are exceptional. My colleague, Dr. Hasmukh Jain, who led the adult trials, presented this data in the ASH American Society of Hematology meeting in 2023, last December. And these were the results for the adult lymphoma and leukemia trials, which again shows you good response rates as they went up the dose level and sustained response rates for more than beyond 6 months in most of these patients. And based on this and the safety profile, is CDACO gave us the approval, ImmunoAct, the approval to market this drug in India. Now, while there may be thousands of clinical trials going on around the world, when it comes to approved therapies, many developed countries also do not have an approved CAR T cell therapy because of the costs involved. So when it comes to approved therapies, I'm happy to say we belong to an even smaller club of countries now with an approved CAR T cell therapy in the market. This club is probably as tight as the space club is in the world. And this was recognized by the President of India, who honored and dedicated this therapy to the nation in a function at IIT Bombay, along with Dr. Rahul Purwar, my colleague Hasmukh Jain, who led the adult trials, and the teams from all of us. So where are we now? Having established this, what I see now is that these are platform technologies. Once you have the capacity to do one from end to end, you can do the others. And this was always our aim. Our aim was never to stop at CD19 to begin with. Now that this one is out in the world, we are both working together and independently now, having created an ecosystem on the next line of cell therapies. These are some of snapshot of some of those which are in the pipeline, some of which Dr. Rahul and I are working with our partner on this established partnership together, and some which we are working by ourselves with various principal investigators at TMC with the help of this cell and gene therapy GMP facility, which has been created at Tata with the help of funding from DBT BIRAC. So this brings me to my last thoughts on this, that having created the ecosystem for team science, cell and gene therapy is really team science. I'm here, a clinician sitting in front of you who doesn't know anything about vectors or how to make them. But for that, you require engineers like Dr. Rahul Purwar and biologists like that. This is a team science. You require everybody. Because we were able to start with a clinical question, we got here fast. We wanted to solve a problem in the clinic, and that's why you got here fast. We have to all work together. It is the key to working in team science events like this is collaboration, and having the entire ecosystem from R&D through manufacturing, through clinical trials, commercialization. Now the last part that we have to work on is to ensure that the model that we have created, that every last patient is able to get this therapy, because even at 40 lakhs to 45 lakhs, which is one-tenth of the cost what it is in the US or in Europe, most of our children and most of our patients will still not be able to afford it. So we need to work on this. And this is where everybody comes in together, academic institutions, government policymakers, international partners, industry, investors, and regulators. All of us, we need to work together to make this and the next lot of cell and gene therapies happen at the fast pace in India. For this, from our side, we are having these educational workshops and conferences on advances in cell therapies. We have had 3 of them. They have been hugely successful. I invite you to come for the next one, which is 7th to 9th March 25. This is probably the biggest cell and gene therapy event in our country now. Ultimately, last thought, technology must benefit patients. And this is, I'm happy to share with you the small interaction which one of our first patients of the phase 1 trial, who is now nearly 2 years plus beyond receiving her therapy, Ishwari, interacting with Emily Whitehead, who was the first girl who got CAR T cell therapy in the world. They had a beautiful interaction with their families and have promised to meet up because Ishwari, just like Emily, loves makeup. And though Emily has begun her freshman year in University of Pennsylvania, the same place where she was treated, she dreams of opening her own salon someday. And I hope that it is more patients like this who will be able to benefit with these therapies down the line. My acknowledgements are many, but I will stick to the important ones, mostly the team members, and of course, Dr. Rahul and the entire team from IIT Bombay, and now ImmunoAct, which is a huge baby of its own. And of course, our funders, that is mostly been Tata Trust and DVT Biorac National Biopharma Mission, which funded a huge part of our facility and the trials that happened after that. I thank you for your attention.
How do you get previous slide? I'm not very familiar with the racing. Oh no, that is tag. Can you go back to? Yeah. So good morning. First of all, I would like to thank the organizers for inviting me and to be part of this esteemed panel. So after hearing the such an inspirational presentation by Dr. Narula about how some of our innovations are really helping patients, I would like to introduce our company and the journey that we have had over last a decade or so. And maybe at the end, I'll talk about some of the challenges that we are seeing and what could be the way forward. So to introduce our company, we are a wholly owned subsidiary of Dr. Reddy's. We are mostly based in Bangalore. We've been around since 2002 with about 450 scientists. I think some of the track record that we are extremely proud of is about 20 innovative drugs that we have discovered are in global clinical trials, out of which about 10 we are leading the clinical development. We've been able to do this in a business model which is sort of self-sustaining. We started as a company that focused on collaborative drug discovery. We worked with a number of companies shown on this slide, including many of the large pharma. These include Novartis or AbbVie, J&J, Sanofi, many of these companies. We worked with them. We identified novel molecules and they took it to clinical development. I think that helped us in a couple of different ways. We could establish as a drug discovery setup in India, and we could also become self-sustained. As a result, we started our own programs. That's what's shown on the left, our own proprietary program that we started. So what we are currently doing is we start the program on our own, we generate IP, then part of the assets we license out to pharma partners, whether it's US or Europe, use the revenue to advance some of the remaining assets on our own with the idea of eventually commercializing some of these products on our own. And we've been quite successful in our licensing model as well. The companies that I've shown you on the bottom right, these are the companies we have licensed out the assets that we have generated at origin. So in terms of the novel therapeutics, initially like many other companies, I think as Ajit was highlighting, small molecule was the focus for many other companies. We had a very good, strong structural biology team. We thought maybe we will focus on soluble proteins. I think if you focus on cancer, being a oncology focused company, we said we will focus on kinases. As you know that kinases play
a major role in oncology or in cancer progression, in survival, as well as the proliferation of cancer cells. We worked on a number of kinases, utilizing the in-house ability to solve the structure of these proteins and design the compounds, having a library of our own fragment-based approaches. We came up with a number of molecules. I've shown some of the examples. For example, we have imavosertib, which is the IRAK4 inhibitor, still the only IRAK4 inhibitor anywhere in the world that's in phase 2. We have a CDK7 covalent inhibitor in phase 1. We have a DDR1 SIK2 in phase 2. This we are doing on our own. We have a SIKJAK that we discovered together with Asana. That time we have Nitin Asana, that's in phase 2. Another one, BRAF PA3K in phase 1. Number of clinical candidates we were able to identify. So going forward, we thought we should also look at some of the newer modality. I think now we know that with targeted protein degradation approach, we can not only modulate the target, we can simply eliminate them. We can degrade the target of interest. Essentially, this is done by using a bifunctional molecule where one end of the molecule binds to the target of interest, other end binds to the E3 or the ubiquitin proteasome degradation system. As a result, the protein of interest is degraded. So we can completely eliminate the bad protein in the context of cancer. We've been quite successful. We came up with a differentiated platform, which we call is directed neosubstrate degrader. These are much smaller. So what's shown on the left, you see that typical bifunctional molecules tend to be relatively large. It's much difficult to optimize them, especially to make them orally bioavailable. With our computational and modeling approaches, we've been able to come up with small molecule degraders. These are significantly smaller. Essentially, they are designed glue degraders. With this, we've been able to do, essentially in our approach, we take the cooperativity between the target of interest and E3 ligase. As a result, we can get paralog selectivity, which is not possible with an inhibitor approach. We can also go for a something that goes after multiple closely related protein. We have two molecules. For example, we have a selective SMARCA2 degrader. We just got the IND cleared by US FDA. We have a pan-KRAS degrader that goes after all mutant forms of KRAS, whether it's G12D, G12D, G12V, G12C. So essentially, we see it's an exciting project. We believe that it has a potential in a number of different cancers. So we are able to start our IND enabling studies with this degrader. And we also had some really interesting publications. We have a publication in Nature, and recently, just last month, in Cancer Cell, where we show that with our approach, we can come up with a degrader that's much more differentiated than any other degraders from any companies in US or Europe. So the next platform that we worked on, as you know that in the oncology space, immuno-oncology or the activation of our own immune system is becoming one of the most important way therapeutic approach. In this case, you know that there are PD-1 antibodies or PD-L1 antibodies that have been highly successful. In fact, if you look at the cancer drugs, PD-1 antibody, Keytruda, is probably one of the most successful drugs. Last year, they sold more than $25 billion. We thought, unlike antibodies, we will come up with a small molecule that's orally bioavailable. So we came up with the first small molecule PD-L1 agent that also targets a closely related protein called VISTA. So this is analogous to kinase inhibitor. When you make a VEGFR inhibitor, for example, it also inhibits FGFR or other closely related target. So we have a first-in-class small molecule oral agent. We have CA170 that's in phase 3. We have a CD47 small molecule, again, first-in-class small molecule immuno-oncology agent in phase 1. We are about to start phase 2 with that agent. We have a TIGIT molecule, again, that's a first-in-class orally bioavailable checkpoint antibody molecule, PD-L1 TIM3 VISTA. Again, I think even in immuno-oncology, especially making them orally bioavailable, that's where I think we made some difference. So similar to most of the other companies, we said we also need to work on biologics, antibody-based therapeutics. When you talk about antibody therapeutics, probably there are three major mechanisms of action we can exploit. First one is simply to take advantage of their natural properties, such as either to block the target of interest or antibody-mediated cell killing, essentially using the monoclonal antibody, naked monoclonal antibody. We can also use antibodies to bring in two different cells, whether T cells, NK cells, macrophages with bispecifics. We can also use CAR-T, as Dr. Narula talked about. We can also look at antibodies for delivering cytotoxic payload in antibody drug conjugate or ADCs or radioconjugates. We also have programs both in plain monoclonal antibodies such as SIRP antibody. We have an ADC program that's ongoing. These are all in preclinical stage. So we also started looking at bispecific antibodies. We believe that I think in the recent past, bispecific antibodies are attracting a lot of attention. I think the most probably widely used application of bispecific in the context of cancer is bridging two cells, whether it would be T cells with tumor cells or NK cells or macrophages. I think probably one of the biggest success story in just the last few days we have heard is an antibody called Ivanusimib. This is from Akeso Summit, Akeso, a Chinese company. So this is the bispecific antibody that targets PD-1 immune checkpoint protein within the tumor milieu, as well as it also binds to VEGF, which is a very strong immunosuppressor protein, and also being an anti-angiogenic protein. So what they have shown is in a head-to-head study, this is a Chinese study, Chinese phase 3 data, compared to Keytruda, as I said, it's a $25 billion drug. This drug was able to show better median PFS as shown in the table on the right. PFS was about 11 months compared to 5.8 months for Keytruda. So I think it's possible to take advantage of the local clinical context, do the clinical trials locally. In this case, they demonstrated that in a Chinese clinical trial, and this has made a huge impact. Right now, everybody believes that this could be the next biggest drug. So I think we should also target making these kind of bispecifics in the Indian context. So we are working on a macrophage bispecific engager that's in preclinical stage right now. The idea here is with this bispecific, we can bring macrophages to tumor cells and eliminate tumor cells. So we also started with an antibody capability that we have, CAR T. So unlike the CAR T that Dr. Narula talked about, we are going after multiple myeloma. So this is a BCMA targeted chimeric antigen receptor, CAR T product. Idea here is we can go after multiple myeloma that selectively expresses BCMA antigen. So we are currently in phase 1. We are dosing patients, and this study is ongoing right now. And this is an India-specific study, but parallelly, we are also developing allogenic cell therapy that would be much more universally acceptable because it could be an off-the-shelf therapy where we can simply make a large batch and dose large number of patients anywhere in the world. So we got into CAR T mainly because we already heard that it's quite expensive therapy. It could cost as much as, say, $500 million per patient. Definitely, even if it's a 40, 50 lakh treatment, it's probably not affordable for many of the patients in India. What we saw was with the current generation vectors, current generation process, there are probably a number of improvements we can bring in. Specifically, if you understand the biology, we can probably manufacture at a significantly lower cost. What we believe is, unlike the traditional 10 or 12-day manufacturing, we can look at the rapid CAR T, manufacture within 3 or 4 days, where you can still retain the fitness of the T cells. That would reduce the number of various media components. It can reduce the cost significantly. Parallelly, we believe that there are opportunities for Indian companies for various consumables as well as raw material that can go for CAR T production. For example, cytokines such as IL-2, which is quite expensive to make CAR T cells. If we start making those kind of raw materials in India, I'm sure we can significantly bring down the CAR T therapy in India. So we believe that in India, we can leverage the economies of scale and bring down these therapies, make them much more affordable. If we can make it less than 10 lakhs, then I'm sure a lot more patients in India can benefit from these kind of cutting edge therapies. And with the next generation, what we believe is instead of viral vectors, can we look at non-viral vectors? Can we use of allogenic off-the-shelf therapy? And also eventually the goal would be instead of cell therapy, can we simply administer the vector? It could be viral vector, non-viral vector to patient, generate CAR T within the patient itself. That would be much more affordable to patients. I think we are looking at some of those aspects as well. And also we believe that in the Indian context, there is a commercial efficiency opportunities. We can attract medical tourism if we can make these therapies much more cheaper, and if we can ensure the quality, I'm sure we can bring in patients from across the world. And if we can convince the payers in the Western geographies I'm sure that will benefit India as a whole. With that, I think finally I would like to share some of the thoughts. We have been developing number of these therapies. What I believe is there are three key pillars that are essential to work together to make sure the innovative therapeutics ecosystem works very well in India. First of all, I think we need to have a very good talent and infrastructure. Unlike, say, IT or the e-commerce, we need highly complex and extensive infrastructure that's needed. What I believe is if we can focus on creating centers of excellence, we can strengthen the state of art facilities, we can foster collaboration between academia and industry as well as government, and we can expand clinical centers where we can do early phase trials with very good safety management system. There should be patient registry so that we know that where the patients are available when we start clinical trials. But probably the major bottleneck or the major hurdle for most of the companies in India is the funding. Obviously, BIRAC is doing a very good job in some of the initial funding, but definitely we need to scale up this funding for drug discovery. What I believe is some of the other initiatives such as preferred track slabs, patent box concession, increase it from 6 to, or bringing it to 6 to from 10%, increase tax exemptions, low interest loans, innovation bonds or other funds. But what I believe is maybe the role of the government should be, if you look at the cost of development of any innovative therapeutics, it could be as big as $2 billion. I don't think we can think of funding a program with a 2 billion investment in India. But I think if we can think of somehow funding it to clinical proof of concept, maybe early phase 1 or early phase 2, demonstrate that a drug is active, then I'm sure we can bring investor from not only India, from also from US or Europe. I think that's what we need to focus on. If government can support taking the molecules up to clinic, maybe generate some human data, that's where the inflection, value inflection happens for any program. So that's what we need to focus on. How can we really fund these kind of programs from the government, from investors, private angel investors, and others who can fund those, think of those things that will be very helpful. So that's where what I believe is one of the unique advantages that we have in India is the large patient pool to be able to do clinical trials a lot faster. We have very good clinical centers like TMH or other clinical centers. I think we need to fully leverage that, the advantage that we have. If we can take the molecules to the clinic efficiently, make sure that the drug has the potential in the clinic, as I said, that's where the value inflection happens. We can much more easily seek the investment from various parts. So that's where I think the role of regulatory comes in, not only for eventual approval, but making sure that our molecules are taken to the clinic, evaluated in the clinic in an expedited manner, and make sure that our guidelines are harmonized with international guidelines, and making sure that we augment the regulatory capacity and capability, and then making sure that there are consistency. Obviously, the Indian regulators have long worked on the generics, but innovative therapeutics is relatively new area. So if we can strengthen some of those ecosystem, I think I'm happy to see that Dr. Vishala is here. So we need to work together, make sure that we generate that critical clinical proof of concept data. I think if we can do it, it will address all our funding issues, talent issues, infrastructure issues. I think these are some of the thoughts that I wanted to share you. Again, thank you for your patient listening. Thank you.
This is back and forward, right? Wonderful. Yeah, yes, please. Yeah, sure. This is the wrong one. Aparna ji, this is the wrong presentation. ठीक है, that's okay. No problem. I'll manage. I'll manage. Leave it, leave it. Very good morning to all of you. Let me begin by thanking BIRAC, Dr. Aparna and Dr. Ajit for inviting me to be part of this fantastic session. I'm going to change track a little bit and talk about how we are using artificial intelligence to discover and develop drugs, mainly for a disease called Duchenne muscular dystrophy. I have multiple affiliations, so I've put all of my affiliations on there. Today I'm representing Pepris Technologies,
which is the AI ML company. So at Pepperidge, we basically use artificial intelligence to come up with either new drugs or drugs which are already approved. We find new uses for them using our AI algorithms. So imagine a large pool of chemical molecules, all from various different chemical classes. Each of them can be represented in very different ways, and our algorithm uses a variety of those representation modules to actually make them into different subsets based on their affinity predictors, their ADMET properties, the physicochemical properties. We can work with small molecules, natural products, and also peptides. And using our algorithm, we are also able to de novo predict some of the new molecules as well. So from all of this, what the algorithm does is basically creates a semantic space which we then use to identify molecules that will work on a particular target. So we start off with a curated library of about 300 chemical molecules, and through our algorithm, we iterate this multiple times to end up with a small number of molecules that we could actually put into the wet lab testing. Using our AI, we do a variety of activity predictions, a variety of molecular property predictions, which are related to both the safety and the toxicity of the molecule. We run a repurposed screen on a majority of the FDA approved molecules, and we also have a pool of about more than 1,000 proteins, human proteins, which we use as part of our screen, where we could not only predict the off-target effects of the molecule, but also identify novel uses for existing drugs. And I will talk to you about one of those today. But before I go into the molecule itself, I want to talk a little bit about this disease called Duchenne muscular dystrophy. Dystrophies as such affect more than 1 in 4,000 children across the world, and Duchenne muscular dystrophy in particular affects more than 5 lakh children in India. The disease itself causes progressive weakness of the muscles and loss of muscle mass due to mutations in a gene called dystrophin. The diagnosis of this disease happens when the child is about 2 to 3 years of age generally, and the lifespan of this disease for the boys who are affected by this disease is up to 21 years. So this is a devastating disease, a progressive devastating disease, which not only affects the children, but also affects families in a large manner. All of this is also complicated by the cost of treatment and the access to some of these treatments, especially in countries like India. The standard of care is a steroid, deflazacort, that is used worldwide. It started off with a price tag of close to about 275,000 US dollars per patient per year, and now has come down to about 30,000 thanks to the generic space. You have a variety of antisense oligonucleotides and gene therapies that are also available, but those are all available in the West. None are approved in India today. The latest addition to this is givinostat, which is a small molecule pan-HTAC inhibitor, which was approved in March 2024 across the world. This has a price tag of about 700,000 US dollars per patient per year. Imagine the kind of cost that is required to treat a patient who's progressively going down over the course of maybe 15 to 20 years. So what we are working on is looking at a strategy where we can restore the dystrophin expression, help the muscle regeneration, and also increase the compensatory protein called utrophin. And all of this in synergy will probably help the patient and improve the quality of life for the patient. One such target that we've been working on is a aryl hydrocarbon receptor, which is involved in the upregulation of utrophin in the muscle cells. This was proven by a molecule called ezetromib, which failed in phase 2 clinical development, mainly because of pharmacokinetic properties. We now have identified, using our AI algorithms, a novel use for an existing drug which is known to target a very different mechanism in the literature. but we have found that molecule to target the aryl hydrocarbon receptor using our AI algorithm. So we started with our pipeline of about 300 million. We identified 33 novel molecules which were predicted using our AI, and one novel molecule, which is one repurposed molecule, which was predicted using our AI. We put this through a series of in vitro and in vivo screens, a biochemical assay for the aryl hydrocarbon receptor, where we showed that these molecules, 6 out of the 33, and the one which is a repurposed, working very well on the target, and then moved on to a cell-based assay, where we were able to show a physiological increase in the expression of utrophin. We followed this up with an assay in the zebrafish model for Duchenne muscular dystrophy, and then onto an animal model in the mice. We believe that the AI tool has actually done a fantastic job of predicting the molecules, and we found that 6 out of the 33 actually perform better than azithromycin in all our assays. We focused then on only the repurposed molecule to move that forward into development. At this point of time, as an organization, we managed to raise our first round of VC funding from VCs in India called Speciale Invest, which was a 1 million round. And this funding helped us to progress the molecule further into development. The molecule is called PEPAR-112. We have obviously not disclosed the name of the drug for proprietary reasons, and this has now been patented globally for a new method of use for Duchenne muscular dystrophy. This is a FDA-approved drug available all over the world as a generic. It is found to be safe in a pediatric population, which is of essence when we are dealing with a disease like Duchenne muscular dystrophy. It can be easily formulated not only as a tablet and an oral suspension for pediatric use, and has a fantastic PK profile which enables it for repeat dosing with a long half-life. Just a comparison data of what our molecule looks like in the reporter assay for the target, which is the aryl hydrocarbon receptor. Azithromycin in this assay has an IC50 of about 4 micromolar, whereas our compound has a almost equivalent or better IC50 of about 1 micromolar. In the zebrafish assay, you can see C1, C2, C3, which are the control animals which have just been treated with DMSO, and the 112, which are 3 different groups there, a total of 68 zebrafish that were part of this study, where you can see a significant improvement in the motility of the zebrafish, which are of DMD nature. So based on this, we can see that there is a significant improvement in the muscle functioning of the zebrafish. Based on this data, we then moved into a mouse model of muscular dystrophy called the D2-MDX mouse. These mice develop progressive muscle weakness and characteristics similar to that of the human disease. On the absolute left, you have a parameter called the grip strength, which is basically just able to hold something and pull on and hold on to it. In this, the DMD control animals, you can see, perform worse than the normal animals. And when we treat them with our drug, there is a significant improvement of almost 60% in the grip strength. We've also done something called as a hanging test, where we allow the mice to hang on to a thin wire. You can see that, and this is done for about 2 minutes. The wild type mice hang for almost the entire period, whereas the DMD controls are barely able to manage to hold on to the wire. When we treat them with our drug, you see a significant improvement in their ability to grasp the wire and stay on with it. The final marker that we did was creatine kinase, which is a marker for muscle damage. You can see the wild type on the left, which is a very tiny bar. And in DMD controls, there's a significant increase in this marker in the muscle tissue. And when you treat with our drug, you see a significant reduction in this marker, which is responsible for the muscle damage. So based on all of this now, we have a response which was predicted by artificial intelligence, which was proven in the cell lines, which was proven in a biochemical assay, proven in the zebrafish model, and now has shown efficacy even in the animal model. Based on all of this, we plan to do a dose response study for this molecule, either alone or in combination with the standard of care, and then move on to directly a phase 2 clinical study in India, obviously with guidance from the regulators, and see how we can use this molecule as an alternate, not only for the steroids, which have significant side effects, but also as a mainline therapy for these patients for which there is very limited treatment that is available in India. Thank you very much for your kind attention.
Good morning, or whatever is left of it, I guess. Um, I first would like to thank for the projection. I would like to start by thanking the organizers for the opportunity to be part of this excellent panel. And I have this unenviable task of matching their excellent performances so far. And I have to admit right out of the outset that I will not be able to match their performances, especially what we have seen is an excellent exposition of what Indian science can do and how we can utilize it to the advantage of not only Indian patient population, but also patients at large across the globe. So what I intend to focus today is really what kind of an asset we need to generate. That's something that will have far-reaching implications and not applications confined to Indian populations, you know. And I work for Sun Pharma Advanced Research Company, and our focus is primarily in oncology. Initially, the focus of the company was for small molecules and the drug delivery systems. But last 5 years, we have been emphasizing biologics as therapeutic module, if you will. And we have initiated efforts, and it took us 5 years to get started crawling first, and then now we are at least able to walk, maybe not run as fast as what Dr. Murli has been able to do in his organization. But our current focus stays on biologics. You know, that's my personal preference and my personal background from my past life. And so I would focus on what kind of innovative products we need to generate or an asset. I shouldn't even call them products because they become products after justification of their utility value. Okay, so what is to be expected out of new generation products? I would personally emphasize that innovation is very critical, which leads to creating novel therapeutic entities or putative therapeutic entities. We clearly need advantageous differentiation. And what I mean by that you have multiple therapeutic options available to patients, right? Intellectual scientific sophistication of your therapeutic at the level of patient is probably not as significant as the clinical benefit conferred by the same entity, right? So keeping that potential to generate clinically differentiated therapeutic activity in mind, when you set out to assemble your therapeutic entity, in this case, a biologic, you need to take multiple factors into consideration. Developing a me too product, which may be a different sequence, patentable, but not necessarily advantageous in terms of conferring greater therapeutic benefit to the patient, you are less likely to get serious traction in clinical utilization of such an asset. So clearly a differentiation element will come handy, which is automatically going to translate into greater therapeutic activity, may it be greater efficacy or greater tolerability. Both are two different sides of the same equation a therapeutic has to deal with. And we often have in biologics the wrong belief, I would say, that you make your own antibody. As long as it has a different sequence, you may even get it patented. But unless you have an ability in the asset itself to showcase differentiated therapeutic benefit, such a patentable asset is not likely to get the same traction. And I made that point earlier as well. So mere difference in the composition of matter or mechanism of action for that matter is not sufficient to sustain commercial viability for such an asset. And again, it comes down to superior clinical efficacy and safety over the current standards of care. And again, we have always been impressed with one important tenet that all we are providing is treatment option for patients, especially in cancer research. There is no promise of a treatment being curative. We enter into the arena with a curative intent. But in more often than not, you end up developing a treatment option that patients can utilize should all other options fail. And it's an uphill struggle in order to showcase the potency of your drug and the applicability of your drug in a particular disease indication, in oncology indication. So our effort has always been to find such an asset or a target structure, target antigen, if you will, for a biologic that will have very broadly encompassing therapeutic impact. And in that, specifically developing therapeutic against an antigen that is expressed on 10% of handful tumors is probably not going to be commercially viable. It may yield efficacy that will be able to help those small number of patients, but the investment needed in order to make that dream realized is not going to be sufficient to sustain the commercial viability. And so you have a breadth of applicability of an asset. You have novelty, has to be a novel entity. And you need to have a clear-cut advantageous differentiation. And I'll come to that in my next slide. So new generation of therapeutics must aim for being transformative with a breakthrough status. We have often used these words being shown. Certain products have received breakthrough status or are regarded as transformative therapies or a game changer or being fast-tracked for regulatory approvals based on obviously the clinical studies that are showing such a promise in a therapy. But we need a therapeutic. The aim should be create a therapeutic that can rewrite the entire therapeutic spectrum or landscape for that particular indication. And I come to the antibody-based therapeutics because myself being an immunologist, my thoughts gravitate towards use of antibodies as therapies. We've seen monoclonal antibody revolution at the end of last millennium when we have Herceptin and Rituxan got approved. And since then, till today, more than 100 antibodies have received regulatory approval for various indications. Not all of those are for oncology, but for a spectrum of diseases, the antibodies as therapeutics are no more a novelty. It's assumed that you will have a biologic therapeutic available for certain diseases. Then came antibody drug conjugates. And I'm particularly very close to this area. I've had the opportunity to work on the first two calicheamicin-based ADCs that got approved, Mylotarg and Besponsa. They both target hematologic malignancy. One is for CD33, Besponsa is for CD22, expressing hematologic malignancies. And I'm very pleased to see that Mylotarg has ascended to the frontline level therapeutic in combination with the current standards of care for AML. And so antibody drug conjugates went through a lull period because of the lack of efficacy seen with multiple number of ADCs seen. And at the same time, you will see that immune checkpoint explosion was happening with Keytruda and the antibodies targeted to PD-L1, they have really changed the therapeutic landscape. So those therapies can be considered as truly transformative in their mode of action. But more recently, I think ADCs have seen resurgence in terms of application of antibody-targeted drug delivery as a strategy in form of antibody-drug conjugates. And you will see that it sort of swings from one end to the other. And what has become realized is that you have to factor in potency and tolerability as important determinants in that consideration. I'll give you an example of calicheamicin. Is a very potent cytotoxic drug that was used in Mylotarg and Besponsa, but tolerability is always an issue. And one of the common denominators for most ADCs is the hematologic and hepatic toxicities, because those are the two organs that handle those potent ADCs, regardless of the antigen specificity. And because of that, there have been thrombocytopenia, neutropenia as common denominator toxicities. You have hepatic enzyme elevation, and in some extreme cases, even veno-occlusive disease that was seen with some of the early studies with Mylotarg. But people have learned to overcome those difficulties and utilize the firepower of Mylotarg for AML, which is one of the most difficult diseases to treat, based on what I hear. So we have resurgence of immune checkpoint inhibitors, although other checkpoint inhibitors other than PD-1, PD-L1 have not really shown the same promise that the concept really holds. So there is PD-1 and PD-L1 still rule the immune checkpoint stage at this point. And then as Dr. Murli pointed out, the bispecificity is an important consideration. Monospecific antibodies will have difficulty getting serious traction at this point because of the technologies that have been evolved to handle bispecific or even trispecific components in your therapeutic application. And that allows you to create, you're hitting multiple birds in one slingshot, if you will, if you use that analogy. You have what would take 3 different therapeutics to get the same level of effect. You have the capability now to punch in that firepower in one single therapeutic. And that is where our focus has been. And you have obviously participation of the cells of the immune system in terms of T cell engagers or NK cell engagers or macrophage engagers, using the appropriate biologically tuned entity. You need to have the recruitment capability as well as the tumor targetability as a built-in component. And that's where the bispecificity is important, where one specificity is for the tumor, the other specificity is for the effector cell that you want to recruit in the cell-based therapeutic application. And then you have CARTs. You heard beautiful presentation from Dr. Narula about how the CARTs can be taken advantage of. Again, it really, CARTs are something that we initially thought of getting involved in, but there were various factors that discouraged us from doing that. And I'm very pleased to see from Dr. Murli's presentation that he's been able to accomplish, get involved in terms of developing CARs as an alternative therapeutic applications. So we have been emphasizing 2 different aspects of it. First, you understand tumor cell biology, and then identify the impact areas that really influence the tumor growth. If you can develop biologic strategies that will interfere in those functioning of those important features by attacking the nodes that are critical for tumor growth, I think you can create a bioactive entity. It doesn't guarantee that it will have the opportunity to outperform the standards of care. And at the patient level, you may have a fancy biologic created, but if it doesn't give you a high therapeutic index and sustainable clinical activity, it's probably not likely to be acceptable in clinical community. And its utilization and by extension, its commercial impact is going to be significantly lessened. So what we have been doing, I'm sorry, my apologies for not showing any slides, data slides that I could have included at the last minute. I didn't. Um, we are creating bispecific or trispecific molecules that will impact not only the angiogenesis inhibition as well as immune checkpoint inhibition as well. And in addition, you have tumor targetability as part of the three-prong approach. So one important advantage that has, and in that you will see all these bioactive entities preferentially accumulated in the peritumoral microenvironment, which may not necessarily happen if the target is a moving target like VEGF or PD-1 for that matter. And it's predicated on the understanding or belief that all tumors are somehow going to make sure that T cells pass through to be benefited from it. So in that regard, I think the new era of bispecific and trispecific functionalities is here, and it's not going to go away. And future therapeutics will have to provide firepower that is brought by individual specificities and the functional impact realized by them. I think I will stop here, and thank you very much.
Oh, they don't have a PDF reader. A PDF presentation.
PDF
Um,
कौन सा वाला आपका
last
ये
was Hello, everyone. My name is Sanatan Upmanyu. Um, I'm the head of data science at Invenia GmbH, based out of Switzerland. We have, it's been a pleasure being here with all the folks here. It's great hearing from what they have done so far and what they have achieved. I would like to focus a little bit different in terms of how AI is helping at each step of drug discovery. Before that, a little bit about me and my company. We have been there for quite some time. We have changed names, but we have already marketed one particular drug as a sister company, and another company we have already phase 2 trials ongoing in atopic dermatitis and urticaria. That's what we are focusing on. We have headquarters in US, Switzerland, and Bangalore. And we have several collaborations with pharma companies, helping them in drug discovery, hypothesis generation, and so on and so forth. So accelerating drug discovery with technology is something that is essential, if not mandatory, because we have to understand how complex biology is. And we have 20,000 human genes that are interacting with each other and also are affecting the cells and organs and so on. So it's a huge knowledge graph of different things trying to make us work as human beings. So understanding what would fail, what would not fail as early as possible is crucial to develop better therapeutic entities. Enhancing clinical success is something that we focus on. Capturing innovations early, that is very crucial. Clinical validation, we have over 10 programs in development with our clients that have gone into one or the other stage of clinical development. And we also have an NDA approval that I just mentioned. Uh, One of the key aspects is once we try to understand a specific disease, we focus primarily on the clinical endpoints, and then we track back that how the biology or AI can help understand or identify better targets, and then create hypotheses from then forward. So that in the end, we have alignment with clinical endpoints, what are necessary for a specific target in a disease. So in terms of drug discovery, um, we have been talking about different type of entities. So novel molecule generation, drug design, target identification, and hypothesis generation is something that we do on a regular basis for our clients. And it's quite crucial to understand that all of these things are very complicated and requires a lot of expertise from our folks from life science and biotechnology background. Not just the AI and software or machine learning aspects. Because asking the right questions as a biologist is crucial to give right results as early as possible. In terms of preclinical research, we have been focusing on drug repurposing, what we started with and we do till now. That's a very lucrative area where there is quick opportunities that could be identified, and it's very interesting to see that already approved drugs could be used into other indications with either change in formulation or adding some molecules here and there to improve the efficiency and help patients as much as possible. Clinical development is something that is very crucial, as every one of my colleagues focused on, and we are taking a different route to that. Although there are a lot of patients in India, patients' lives are important, and we are currently focusing on developing digital twins and simulating clinical trial based on electronic healthcare record data and patient data to simulate individual patients and see that how they could potentially behave in a specific therapy. These are very complicated models. We have to develop Boltzmann machines for individual patients to understand how each patient would behave with different background and different disease biology and what stages those patients are. So all of those data points goes inside, and then we are able to come up with these kind of digital twins. And obviously the health technology outcomes are crucial for these simulations to understand that what kind of drug in what kind of phase and what kind of disease would be better for an end objective of, say, suppose launching in Europe versus India versus US. So all of those things are crucial and need to be baked in together. Post-marketing surveillance is something that we do very rarely, but we focus on actively doing real-time assessment of drugs by understanding what kind of uh, approaches different payers are taking towards a certain drug. These are some of the tools that we use in AlphaMeld. That's our leading platform that we have built in different stages of drug development process. As you can see, we have network designers, dashboards, automated report generations, ChatAlphaMeld, which is helping researchers giving right answers as soon as possible, which has way higher accuracy and almost no um, almost no hallucination associated, and we get real-time data associated. Chattelfameld also has other generative models associated that we are currently developing that will help in uh, drug design and drug optimization aspects. Um, we boast a lot that the amount of data that we have. So just from the starting, we have already processed 1 billion sentences of scientific literature, including clinical trials and patient data, and we are able to understand how the biology works with different type of entities and how they correlate with each other. One of the key examples that I would like to focus here is obesity. So if you see, this is how complex obesity and diabetes area is in terms of how they interact, how the genes, cells, drugs interact with each other. So it's humanly impossible to understand what would be the right target and what kind of safeties would be associated with each of them. So in this case, we can see that we have listed some 13 disease symptoms. So there are 1/4 of human genome is associated with obesity, and they have one or the other way connected to the disease. And understanding it from an AI perspective, using deep neural networks or neural networks and right now generative models, combining together is crucial to provide better safety aspects and efficacies associated to potential novel candidates that we want to move forward with. In terms of key takeaways, I would like to focus on transformative impact of AI in therapeutics. That is something that is already happening, and we should move forward with this particular approach of reducing the burden as much as possible on our researcher by doing a lot of AI-based understanding about the target, drug, disease as much as possible beforehand. Collaborative innovation is essential, as rest of my colleagues have mentioned. It's crucial to do collaboration and improve and raise funding as much as possible to give better outcomes. Real world data is something that is crucial that we have understood over last several years. Patient data is very crucial. The expression data associated with patients are very crucial to understand and build the right biology tools to understand how human biology works. In terms of future directions, there is definitely we need to expand beyond what we are doing right now. So the collaboration between the biologist and the AI focused people is crucial to build better tools because one cannot do a good research without AI, and AI folks alone cannot build right tools that will help biologists or biotechnologists moving forward. Development of AI enhanced drug repurposing, that is already a lot of companies are doing, and I think it's a very good way forward because we already know that there are marketed drugs which are safe and can be repurposed into other indications. Regulatory advancement and standardization is something that I would like to emphasize on because this is an area where we would like to make as much standard approaches as possible so as to automate or move forward as much as possible, not just in terms of the actual drugs, but also moving forward AI-based tests that can identify patients quickly as possible. So with that, I will end my presentation. Thank you very much.
Hello? Yeah, thank you. Thank you all the panelists. Most of the focus has been on how do we identify molecules that can go into the clinical trials, and that's where, we have the opportunity to hear from and Dr. Vasala. I think she's going to tell us how we are going to take this forward. an effort of this type, I think it has to be joint, working closely with the regulators, get their guidance on a regular basis, and move the project forward. In that way, we'll be able to bring drugs, novel drugs in particular, to the patients faster. So from that point of view, Dr. Usala, would you like to comment on how various stakeholders can work with you and get your guidance so that we can move the projects forward towards approval? Thank you.
Um, Thank you for the question and having me here for this session on biotherapeutics. A lot has been very nicely presented in terms of the developments in the space of new age generation technologies, starting from MABs to CAR T cells to bispecific and other upcoming technologies. As you all are aware, that the basic legislation in the space of regulating drugs has been the Drugs and Cosmetics Act, and very rightly pointed out, India has been a hub of generics. Indian manufacturers do file a lot of ANDAs today. They do conduct several bioequivalence and bioavailability studies. India had become a global hub for conducting clinical trials, and we were at a point in time doing a lot of global clinical trials. This was a route through which several new molecules could be launched in the country, and by approaching CDSCO for marketing authorization. Basically, the phase 3 trials, a limited phase 2 trials, and now India has grown and the scientists have developed capabilities to go to the phase 1, and including, like we heard today, the AI-based development of drugs and understanding the toxicity or the safety better, even before we take them to the clinic. The Drugs and Cosmetics Act had a Schedule Y, which was enunciated way back in 1988. We worked with it, we used it, and a lot of drugs have been approved using the very same Schedule Y, which talks of the requirements for non-clinical studies, talks of the clinical data requirement, etc., for marketing authorization. We moved on in 2019, and we now have the new drugs and clinical trial rules, 2019. This piece of legislation has several new features. We do have opportunity for seeking waiver from conducting local clinical trial in India if the product is approved abroad, or Indian population do not behave differently in terms of ethnicity, etc. We also have now the accelerated approval pathway, expedited review pathway, the pre-submission meeting pathway, and post-submission meeting pathways. We have also developed various guidelines, more specifically for vaccine development and biotherapeutics, basically the biosimilar guidelines. We are yet to come out with a clear regulatory landscape for the cell and gene therapy space as a chapter to the new drugs and clinical trial rules. And while that is being processed, CDSCO, I'm sure yesterday Drug Controller General was here, he would have apprised you of what we are doing at CDSCO to strengthen the legislation in this phase of biotherapeutics. However, I would say that 90% to 80 to 90% of CDSCO works today are through the online module. We call the portal as a Sugam Online. We also have the medical devices which we regulate, for which we have a separate portal. So all in all, what I would like to say is that there are several works in progress, particularly in the development of guidelines. And as we move forward, we may have more regulation in the cell and gene therapy space, and other biologics as well. However, I would also point out that ICMR has developed, along with CDSU, the stem cell guidelines and the gene therapy guidelines. So these are the basic guidances which the scientists and industry can look to use it. And in case of any specific queries or questions, they can make a submission to CDSU through the pre-submission route, or you can also, we also address your queries through a public relations office where you come and meet the regulatory authority to understand the regulatory pathway. Yeah, this is what I have to say for your question.
Absolutely. I think the fact that various companies can come and meet your colleagues there in the public relation office and get some guidance, I think that has immensely helped, particularly for a lot of startups who are there in the audience. And thank you very much for sharing your insights on how we can work, how we can partner with you so that we can bring the medicines to the patients much faster. Thank you very much. I know you're busy and you have to go for one more session. Thank you. In the interest of time, I have a couple of questions before we request the audience if any questions are there. One is Dr. Narula. It's very nice that you talked about new clinical trials that are coming on CAR-T. But the challenge could be on the manufacturing because each of these are for individual patients, and the more patients we see, we get excited. But I think it's important that we run as many CAR-T therapies as possible. But can you comment on the challenges you have with regard to manufacturing? Is that a limiting factor, and how are you going to address that? And then the same question for you, Murli and Sridhar, if you can answer briefly.
Thank you. I think that question is very relevant, especially for the cell and gene therapy space. There are 2 or 3 unique identifying features of the cell and gene therapy space which separate it out from the rest. One is where the R&D is done, and 2, where the manufacturing is done. And third, that it is personalized. So far, most of these products are autologous. So by personalized and being autologous, it means it has patient-centric. So this is one crucial aspect of it. The second is that where is the R&D occurring? Where, like I said, as in our case, the R&D occurred in 2 academic institutes. That has been the case with Cambria, also the first one. Whereas now you're seeing Origin now coming up with the next. But whereas when I say this to contrast it with biologicals and pharmaceuticals, where almost all the R&D is done primarily in pharma, in companies. So what we are seeing in cell and gene therapy spaces, R&D is also going to come from here. And I think this is one area where the balance will remain between academic centers and companies. The crucial aspect that brings into place where is the manufacturing occurring. Again, manufacturing up till now has been the domain of companies, industry. Whereas, like we saw in our case, manufacturing started in actually academic centers. So now this is a new area for clinical centers and academic centers to be stepping into the domain of manufacturing. And because these therapies are personalized, and they involve living cells and have to be close to the patient, I see this happening for the next to the line therapies also. So this is one of the biggest challenges for academic centers to meet industry level norms, because the norms are the same, is one of the big challenges, and that's where we have been looking for regulatory guidance and have been getting it, in fact, to navigate this pathway. But this is going to be a challenge for the cell and gene therapy space. Once the early phase trials are done, then it needs to scale up. In scale up, there are 2 models. Of course, ImmunoAct, I think Dr. Rahul Purwar would be the right person for that. Industry level scale up of mass production of these therapies, even though they are personalized, is one model. And the other is to go the allogenic route, where they can be produced and manufactured and stored to be used as and when required. So the second model will be more prone to massive scale up, whereas this will be limited by patient personalization. So these are the 2-3 unique aspects of manufacturing for cell and gene therapy, which are going to define the future. And the more we have clear pathways for these, it will be easier for the field to take off.
So I think Dr. Narula touched upon all the key aspects in terms of cell and gene therapy, but clearly we believe that even with the existing technology, existing vectors, existing targets, even autologous, there may be some opportunity, but probably limited opportunity to reduce the cost further. So that's where I think we are looking at various ways that we can bring the cost further down. But I think in the long run, we need to think of, can we really make these therapies affordable? So that's where, as rightly highlighted, allogenic therapy. But eventually, what I believe is it has to be purely vector based. One should be simply able to inject the vector and should make the CAR T or CAR NK or whatever approach that you are going to go after cancer cells. It has to be in the patient's body. Then I think it will be truly affordable. So there are some globally, some trials are just initiating, but we believe that we need to also start thinking about those kinds of therapies. Then I think pretty much gene therapy or cell and gene therapy would be truly affordable. So we are thinking along those lines, but I guess we still have long ways to go to get to there. Yeah, I think in between we need to obviously go with the current technology, look at allogenic. In terms of the other therapeutic approaches, clearly I think we have an opportunity. I think with the BioE3 policies, I think with greater emphasis on biotechnology-based precision biotherapeutics, I think we have a significant opportunity. I think if we can show that we can manufacture these biologics in a much more effective way, we could be a global hub for biomanufacturing. So I think with greater emphasis on whether it's monoclonal antibodies, bispecifics, ADCs, I think we need to start focusing on. I think what I heard was just I was last week in a conference. Uh, Most of the companies, even Western companies, they say that if they work with company who has done it end to end, who understands the discovery part of it, the quality of the output is much better than somebody who would just manufacture it. So that's where I think we all need to start thinking about how do we develop novel biologics? How do we develop novel ADC? How do we develop novel cell and gene therapy? I think if we can start showcasing, I think we will have greater conviction to convince pharma companies to work with us. Then we could, our BioE3 policy would be hugely successful. We could be the manufacturer for even rest of the world. So I think that's where we believe that we need to start thinking. Let's start from end to end. Let's showcase that we can do it from discovery all the way to commercialization.
Yeah, I couldn't agree more with you, Murli. Manufacturing often is not paid serious attention to while you are trying to discover novel entities. And we happen to rely extensively on professional CDMOs to manufacture it on a large scale. And if internalization of manufacturability is not an easy task, we explored that at Spark, but then soon realized that we need to continue to feed that engine infrastructure that you build. And that is not always going to be guaranteed. So unless you open up for being a CDMO service provider for multiple such customers, I think it becomes then cost in effect.
Thank you. Yeah, Dr. Visala, you want to comment?
I wouldn't agree more than what Dr. Murli has just pointed out. These products are personalized medicine, and at the same time, um, these are new age technologies, and the more we learn and the more we know about them in terms of their design, only then can we have consistent manufacturing. So it's all very important to integrate the learnings that is from the laboratory to the clinic,
Yeah.
and then taking back the learnings as a full curve through PV. However, since the process is a product in these technologies, I think there's a lot that needs to be learned and taken forward for consistency in production and manufacturing.
Absolutely, absolutely. Couldn't agree more. Yeah, thank you very much. I had quick questions for Sridhar and then Sanatan. You know, AI is coming to the forefront, and you have successfully shown, particularly for disease like muscular dystrophy, how you can utilize AI. Can you comment on, you know, other projects that you may have for diseases similar to neglected diseases, including including muscular dystrophy?
I think AI is a technology which can be applied across therapeutic areas. So specifically at Peptris, we are looking at working in the rare disease space. Duchenne muscular dystrophy is one. We are looking at FOP as another indication, SMA as another indication. what has happened is since the last 1 year, since we raised our first round of funding and got some positive press, there has been a lot of approach from parent organizations. A lot of these rare diseases therapeutics are driven by parent organizations. And these parent organizations have been in touch with us to kind of see if we would be interested in working on some of the rare diseases that affect their children. And so I think the breadth of rare diseases that we are working on has suddenly increased. But in addition to the rare diseases, we work in the areas of inflammation, oncology, and also infectious diseases, where we have partnerships with a couple of other organizations.
Thank you. So we have worked in past with companies focusing on rare diseases. And one of the key focus is to, again, focus on repurposing capabilities, um, how you can map all the existing drugs to all the rare diseases, and then try to see that which one of them make more sense and would be easier to go forward with. AI would be again crucial in identifying that which one of them is more amenable and where you can get more funding initially to build a proposal and project.
Any questions from the audience? Yeah, please go ahead. Please be brief.
My question is on disease monitoring, particularly treatment monitoring. We are developing a lot of next generation therapies. What about tools for monitoring these therapies? Is there because I do believe that given the cost of these therapies, we need very efficient tools that will tell you whether the therapy is working at the beginning, and also, if possible, to qualify a patient, whether a patient is suitable for a particular therapy. And in fact, I would like to hear from Dr. Narula on this.
We have, yeah, one functional mic. So you're absolutely right. That one of the first challenges when we developed this therapy was how are we going to monitor its response. Uh, as you know, it's a CD19 directed therapy, and the CD19, all cells carrying CD19 get eliminated. And our strategy for monitoring the disease, the flow cytometry, which we use, begins by gating CD19. So now your gating strategy is gone, and therefore now you have to you know the leg which you stood on for monitoring the disease is no longer there. So first we had to we had to sort of finalize our flow cytometry plan. Luckily for us, we were also working with a bilineage T cell engager, that is blinatumomab, which was we were getting through an assistance program, and we could fine tune our gating strategy, and that's published work now from our group in Tata Memorial Hospital, how we are going to monitor the disease. So this was one of the first steps. The next step became flow cytometry can monitor for you disease to a certain level, and it can predict CD19 positive relapse if you see CD19 returning, but you cannot predict CD19 negative relapse very well, and that can occur. So for that, we have to now develop a next generation sequencing strategy to monitor the disease. So this is going hand in hand with the trials, and that work will come out when it is when the trial results are being published. Some of the work which was the strategies, they have already been done. But similarly, just like the challenges in this one, with each new cell and gene therapy that we develop, we are going to face this challenge. We are knocking out the target. So now monitoring the target, it becomes very difficult, especially when you're using the same antigen to identify whether it is gone or not. So for solid tumors, you will depend more and more on imaging, maybe tagging other antibodies. But in hematological malignancies where I work, this is always going to be, challenging and innovative. So it's a very important area, and we are working on it. My PhD student who's in the audience is working on the in vivo monitoring of these cells and what is going to happen to them and their dynamics. So we are looking into this now. And the other area which we're looking into is the manufacturing research. So we're looking at the in vivo dynamics, and we're going to learn from that and get back to the bench and design better strategies for cell and gene therapies to get back to the clinic. So we are trying to do this full circle now at our center. Thank you.
I think there's one more. Please be brief.
Hi,
Thank you.
I'm Dr. Nusrat, founder of Cygenica. I have a question and a request. My question is, you all have explained about the new age therapeutics on CAR-T and other ADC space, but how about genome editing therapeutics and other RNAi-based therapeutics? Where are we at? And my request is for Dr. Murli and Dr. Nitin Damle that you rightly mentioned about end-to-end doing the innovative pharmaceuticals. We startups struggle in getting credibility as we have a novel drug delivery molecule. I would request you guys supporting us in evaluating it or taking some early risks so that we get better credibility for developing other bigger pharma partnerships. Thank you so much.
Yeah. You want to answer that?
Yeah, so maybe your second question first. So clearly, enabling technologies are clearly important for companies like us, whether it's delivery, whether it's a different formulation. So that's where I think companies such as yours, a lot of startups, even academia has a big role to play. I think more than developing therapeutics, I think the academia probably the biggest contribution they can make is work on a lot of enabling technologies that will help us in having the right infrastructure. Even for simple example, for example, cryo-EM. There are maybe only 3-4 places we can get structured by a cryo-EM. Can we have more of those? Because those are the ones, if we have those facilities in specific centers, then we can access them. We will have a lot of people who are trained. So we can enroll talents who are familiar with those technologies. So clearly, I think that's the need of the hour. Enabling technologies from academia, startups. I think we need to work with relatively mature companies. So that's where I think we need to see whether there is a match that we can really take advantage, that we can leverage. In terms of the genome editing, obviously with CRISPR and other approaches, right now the challenge is the delivery. So some of the vector system that we are looking at in CAR-T, they are equally relevant. So we are thinking, can we use the lentiviral vectors? Can we think of new generation vectors, whether it's non-viral or non-lentiviral vectors? Right now, AAV is probably one of the leading vector systems. So we are thinking along those lines. But genome editing probably has a relatively less focus in oncology. That's one of the reasons we are not really so gung-ho about genome editing. But clearly the vector system that we develop for gene delivery has, I think, relevancy for even genome editing. So that's what I would say. Nitin?
I think as far as Spark is concerned, that's not on our scope of activities that we can invest our limited resources in. So we've been focused more on creating targeted therapeutics, biologics, and small molecules as well.
Yeah. Yeah. Thank you, Nitin. I guess, yeah, if you can be brief, thank you.
Yeah, sure. This is Dr. Shubhra Chakraborty. I'm representing VJ Biosciences. It's a startup in Bangalore. So my question is that for the CAR-T, recently, I think the Stanford, what they have found out and the FDA black box with respect to the future of the CAR-T. I mean, my question is that there is a risk and how we see that risk is going to impact overall the CAR-T research and how we are going to overcome it.
So, were you referring to a specific risk?
I think it's not a specific risk. Now, for all CAR-T, there is a black box warning that they
okay, yes, I think I know what you're referring to. So, you're talking about the black box warning for second primary malignancies. So, it's a very timely question because just yesterday, actually, just yesterday, a huge meta-analysis of second primary malignancy incidence was published. Now, there are 2-3 things over here which are crucial. CAR T cell therapies so far have been used as end-of-the-line therapies, and now they are moving into up the line. So in myeloma now, they are reached beyond second line. In DLBCL adult lymphomas, they have reached second line, but nowhere are they first line yet. So we are talking about relapsed refractory situations where the risk of dying in the next few months is very high. And this is the category of patients who are now surviving and in whom you're seeing some of them, we have seen second primary malignancies. So that has to be put into context first. In that context, the risk benefit ratio remains very much in favor. The question that remains to be answered is that these second primary malignancies, whether they are occurring, whether they would have occurred anyway because of other reasons, whether they would have occurred because of the prior chemotherapies which the patients have received, or because of the underlying genetic conditions which make them prone to malignancies, or are they due to the CAR T? And with the malignancy itself is carrying the CAR T gene or not. So now, obviously, the first few, the only malignancy which can be directly related to the CAR T is if you get a T cell malignancy in the patient who has survived the initial indication and now develops a T cell malignancy. Now, this incidence was seen in a few patients. They were reported. There was some, and when about by the time about 12 malignancies or so were reported, the FDA asked for an audit, and on the basis of that, they put a black box warning for all class as a class product across all CAR T cell products. Now, what we are looking at is that in those T cell malignancies, first of all, are they carrying the CAR T or not? Many of them were not. And in those which were, the next thing that remains to be proved is whether the CAR T caused the switch or whether it was just an incidental. It's a T cell malignancy in the cells which also happen to be carrying the vector. So these are the questions which remain to be asked, which are still unanswered. But a large meta-analysis which just released yesterday showed that the incidence of second primary malignancies in the patients who received CAR T were not higher than those which would have been otherwise expected in that same cohort of patients. And it's a large meta-analysis. I would like you to go through it. This was just released, published yesterday, and it is. Since I was here, I have not yet read the full article and all the tables. Otherwise, I would update you on that. But that's where it stands. And the FDA, we asked Dr. Mark the question directly, and he clarified. He says the risk-benefit ratio far exceeds this. This needs to be explored. So it's a black box warning, but we'll continue. The problem will come when you start trying to use CAR T cell therapies in indications which are not otherwise fatal or where alternative therapies exist. Let's say immunological conditions, for example. Now, some of them are severe and fatal where it might be justified, but in others, this question will come up. Can we risk curing from a disease from where the alternative is available and risk a second primary malignancy? So these things still remain to be answered, but now the jury is out and they've come back with a verdict. Yes, in malignancies, we can go ahead with CAR T and should.
Okay, I think, I don't think we have enough time for any question unless we are very brief.
Yes, sir. So, hello, I'm from the Science and Technology Department from Government of Kerala. I have a suggestion for the regulatory bodies related to the microRNA and long non-coding RNA based therapeutics for the rare diseases as well as infectious diseases. My suggestion is have a connection to the first question related to the tools. How are we going to monitor it? The suggestion is if the microRNA and long non-coding RNA are going to be safer phenotypically as well as genotypically for the first 2 years, then I think we should not have any issues related to the patient. Then only these kind of therapies can be helpful for the patients. If you keep on monitoring them for the long period as the Western world does it, then it is not really required. 2 years are more than sufficient to prove that these kind of therapies are safer to the patients, provided that physiologically and biochemically and specifically phenotypically safer to the patients. That can be considered. Then we will discuss it further later. Thank you.
Yeah. Thanks for bringing it up. I think it's very important, but you need to have a discussion with their, PR office and take it forward. Thank you. There may be one. Yeah, please be brief.
Yes, hello, hi, I'm Dr. Yashwant from Minga Therapeutics. So I have a single question. There is a space between antibodies and small molecules. Are there any companies that are working in between? What do you think the future of those kind of like peptides, nanobodies, or that kind of technology? And also, I have a small stall on 80. I request all the eminent panelists to please come and visit the stall. Thank you.
Sure. Okay, thank you. You want to comment quickly, brief?
Sure. So I think peptides and especially cyclic peptides are coming back into the fray, and that's a definite area of interest for Peptris as well. So definitely an area to watch out for.
Okay. Yeah, thank you, Sridhar. I think we would like to conclude now. I think all of you panelists have been working on novel therapeutics for various diseases. One last request with each of you is quite a number of participants here, particularly startups looking at venturing into new therapeutics. So you have one advice for them?
Oh, I am at the business end of. I say we call ourselves the foot soldiers of science. You are the brilliant guys and the people who make new therapies. I am the end user. So if you got something which is likely to work and is moving into trial, we are very interested because there's a huge need, unmet need for our patients.
I think if you believe in a new technology or a new approach, I think we should go for it. I think other things will follow, whether it's funding and other things. So it's having that conviction is more important. I think if we believe in something, things will happen.
Patience and perseverance. Two qualities I think every startup needs, right? Good things will happen.
Yeah, thank you. Thank you, Sridhar.
Your question reminded me of a line in a movie, Field of Dreams, where the advisor says, "You build it and they will come." And he was trying to build a baseball stadium, right, in cornfield out of nowhere, and he succeeded in that. So asset generation is what I would call this particular aspect. And you can build in, as I was saying, that as much firepower as you possibly can, and as the technology allows for it, go for it. I think there will be takers.
Yeah, thank you, Nitin. Sanatan?
Yeah, I think it's very important to understand how you want to go forward. Understanding both biology and AI, at least from my perspective, was key for me. But I think you guys need to understand what is your true calling and then go forward with it. Thank you.
Yeah, thanks, Sanatan.
Yeah.
Dr. Vishala, thank you.
Coming from the regulatory front, it's always SEQ, safety, efficacy, and quality. Keep that in mind. And performance, obviously, only then I think it would be a wise investment for you as a startup or an industry. But I'm sure there are a lot many tools today to identify the risks and mitigate the risk. And regulators always look at benefit versus risk, as Dr. Narula has also pointed out.
Yeah, yeah, thank you very much. Thank you. Back to you, Aparna.
Thank you, sir. So with this, we have come to end of the session, and I would like to give my sincere thanks to all the panelists. And I would now request Dr. P.K. Sharma to please come on the stage and facilitate, sir, all the panel members. So I would like to request to facilitate Dr. Ajit Kamath for moderating the session, sir. Thank you for being here today, sir. Dr. Gaurav Narula from Tata Memorial Hospital. Dr. Murli Ramchandra. Dr. Sridhar Narayanan, sir, from FNPR. Dr. Nitin Damble. Dr. Sanatan Upmanyu. Dr. Anam Visala. Please come. So I would request for a group photo, please. Thank you very much, sir. Thank you. And can you play the video? We have a small video from the startup, so if you can play in the background.
STEM Research is a clinical stage biotechnology company incubated at the Center for Cellular and Molecular Platforms, Bangalore, supported by DBT Biolab as well as prestigious Indian and global healthcare investors. Our first product, iSight RPE, is developed to treat dry age-related macular degeneration, dry AMD, which is the largest cause of incurable blindness in the world for patients over 50 years. iSight RPE is a suspension of iPSC-derived retinal pigment epithelial cells, which are highly potent, safe, and proved to be efficacious both in vitro and in vivo using normal and diseased animals. We are the first biotechnology company in India to obtain IND approval from DCGI CDSCO to start phase 1 to a clinical trials for AMD, and 3 patients have successfully received injections in the last 2 months in AIMS Delhi, Vipin Prasad Eye Institute, Hyderabad, and Sri Ganpati Mitralaya, Java. Besides ophthalmology, ISTEM is also working on lung disease called pulmonary fibrosis, affecting more than 5 million people globally. We have developed a published and patent pending protocol to generate airway and alveolar cells from human-induced pluripotent stem cells. Currently, we are investigating the antifibrotic potential of these iPSC-derived lung cells against COVID-19-induced pulmonary fibrosis in animal models, a project that is funded by COVID Suraksha scheme under BIRAC.
Hello, my name is Dr. Siddhartha Mukherjee, and I'm one of the co-founders of Immunine. It gives me immense pleasure to welcome and start the opening of this company. Immunine is a company that will deliver cellular therapies for cancer to India, such as CAR T cells, T cells that have been genetically modified to kill cancer cells. These have been found to be extraordinarily effective in cancers such as leukemias, particularly childhood leukemias, but until today have not been available in India. This moment makes me extremely proud, and I welcome the team and I welcome the opening of this facility. Thank you very much.